Our topic for tonight is going to be the origin of life, probably one of my more technical talks, a lot of biochemistry and such, but I'll do my best to bring it down to layman's levels. If you want to get the lecture notes for this talk, use that QR code. This is, I think, one of our most important topics in apologetics, the origin of life is, and definitely one of the biggest challenges facing evolutionists today is the origin of life. And for many people, it was one of the things, for many Christians who have come to accept evolution, I think it has been the origin of life and the evolution of humans. That was the two things that caused them to withhold accepting evolution. For many, I mean, we can recognize that organisms have been created with the ability to change dramatically over time. I mean, all we have to do is look at how many dog breeds or cat breeds that we've created in just a few thousand years to see the amount of evolutionary potential that is within God's created order. But to accept the full-blown teachings of evolution, that common descent of all organisms on earth, to accept what they're teaching about the full picture of evolution, for many people required that they come up with evidence to support the origin of life and through natural processes alone, and that humans have evolved from apes. Those are the two big ones. And but I think when you examine those two subjects, you'll still see that they're completely unsupported. I mean, the origin of life and the evolution of humans from apes are completely unsupported. But you know, these for at least the origin of life, these are what they're teaching today are really not new things. Some of these go all the way back since the time of the ancient Greeks. It was believed by your natural philosophers, people that held to naturalism, and the natural philosophers and scientists all the way back since the time of ancient Greeks believed that some forms of life had arose spontaneously on the earth. This is what was called originally spontaneous generation. Today we tend to call it abiogenesis, but even Aristotle wrote a book in 350 BC, shown here, titled The History of Animals, where he talks about spontaneous generation as the way that some species originate from previous living tissue, such as when maggots would appear on rotting meat. It was thought that rotting meat, previous living tissue, could give rise to new forms of life. It was in 1668, Francesco Redi, an Italian physician, challenged this teaching of spontaneous generation in an experiment where he placed meat in various containers, some that were open to the air and some that were sealed completely and others that were covered by a gauze. As he had expected, maggots only appeared on the open flasks containing meat, because flies in that case were able to reach the meat and lay their eggs on it. However, despite this experiment, the belief in spontaneous generation remained strong. And even Redi continued to believe it occurred under some circumstances. The invention of the microscope only served to further the belief. Microscopy revealed a whole new world of organisms that appeared to rise spontaneously in water that had nothing but grass in it. It was quickly learned that to create these organisms they called animalcules, you needed only to place hay in water and wait a few days before examining your new creations under the microscope. And we do this today, actually, to create a culture for observing protists, these various types of single-cell organisms. All you have to do is get a handful of hay and stick it in some water and leave it for a period of time, and all kinds of single-cell organisms will appear there, which are very cool to look at under a microscope. But the theory of spontaneous generation was laid to rest conclusively by this guy, Louis Pasteur. He was a devout Catholic who is known today as the father of both microbiology and immunology. His discovery that most infectious diseases are caused by germs, a theory known as the germ theory of disease, is one of the most important in medical history. He also discovered multiple disease-causing organisms. Discovered and isolated multiple disease-causing organisms. He discovered and isolated Staphylococcus that gives rise to staph infections, Streptococcus that gives rise to strep throat, and Pneumococcus that gives rise to pneumonia. He also developed vaccines for many diseases, such as chicken cholera, anthrax, and even rabies. Well, what he did to disprove spontaneous generation was to conduct an experiment as part of a contest that was put on by the French Academies of Science. They put on a contest for the best experiment to either prove or disprove spontaneous generation once and for all. And this was coincidentally a contest that occurred in the same year Darwin published his seminal book On the Origin of Species, 1859. Well, in Pasteur's award-winning experiment, what he did was he put some meat broth, nutrient broth, inside of this flask. This is actually Louis Pasteur's flask using this experiment. Well, the neck of this flask was bent, the way you see there, called a gooseneck flask, but it remained open to the air. It was not corked or stoppered in any way. And so he took this flask with meat broth in it, and then he boiled it to the point where the meat broth was completely sterilized. And as he expected, nothing grew in the meat broth, even though it was open to the air. Then what he did is he tilted this flask so that the media was able to reach the bend in that neck and then flow back into the flask. And after just a day or two, the flask became cloudy as microorganisms germinated and began to grow, bacteria and fungi, because they had fallen out of the air and had collected in the bend of that flask. So with this simple experiment, not only did it disprove spontaneous generation conclusively, world-renowned experiment, disproved it conclusively to the satisfaction of everyone, but also demonstrated that microorganisms are everywhere, even in the air. Well, despite the global acclaim that Pasteur received from this experiment, evolutionists have continued to uphold the belief. Charles Darwin himself was still advocating for spontaneous generation more than 15 years after Louis Pasteur's experiment. In 1871, Darwin stated this: "It is often said that all the conditions for the first production of a living organism are present which could ever have been present. But if, and oh, what a big if we conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. were present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed." Even though Louis Pasteur had disproved spontaneous generation, here Charles Darwin himself, in a letter written to a botanist friend by the name of Joseph Hooker, was advocating for spontaneous generation. And then almost 20 years after Pasteur's experiment, this guy, Ernst Haeckel, one of Darwin's most evangelical promoters in Europe, continued to advocate for his belief on purely religious grounds. In 1876, he stated this: "If we do not accept the hypothesis of spontaneous generation, then at this one point in the history of evolution, we must have recourse to the miracle of supernatural creation." Haeckel continued to choose spontaneous generation because he did not like the alternative, belief in God. Well, this man, you need to know, you need to understand, was significantly responsible for evolution being accepted in Europe. In fact, he was the first person to introduce the theory to Germany. And even Charles Darwin himself believed that Haeckel's work and promotion led to its acceptance there. And if you understand what Germany was to Europe at this point in history, they were the scientific and intellectual leader of Europe. And so once Germany accepted Darwinism, Europe did as well. So this guy right here, Ernst Haeckel, is responsible for Darwinism being accepted not just in Germany, but in Europe. In 1908, Haeckel received the prestigious Darwin-Wallace Medal from the Linnean Society of London. During the award ceremony, the president of the society read the following commendation of Ernst Haeckel. It says, "Professor Haeckel, a personal friend of Mr. Darwin, who paid more than one visit to him at Down, where Darwin lived, has been the great apostle of the Darwin-Wallace theory in Germany. His advocacy of the doctrine of evolution in his monograph on radiolaria in 1862 first brought it to the attention of German men of science. His enthusiastic and gallant advocacy ever since has chiefly contributed to its success in that country. Mr. Darwin, in 1873, wrote to Professor Haeckel and said, "You will do a wonderful amount of good in spreading the doctrine of evolution, supporting it as you do by so many original observations." Well, Ernst Haeckel was the first person to draw an evolutionary family tree. That included mankind at the top. I've circled it for you at the top. But to fill in the gap between apes and humans, he invented an ape man. He argued that the only major difference between apes and humans was that humans could speak and apes could not. He therefore fabricated a missing link, which he called Pithecanthropus allelis, which means speechless ape man. He had an artist draw the imagined creature, although there was not a scrap of evidence to support its existence. A contemporary in Haeckel's day, a guy by the name of Rudolf Virchow, who was the founder of cellular pathology and for many years was the president of the Berlin Anthropological Society, Rudolf Virchow was scathing in his criticism of Haeckel. For Haeckel to have given a scientific name to a creature that had not been shown to exist was to him a great mockery of science. Well, then in 1883, 24 years after Pasteur's disproved spontaneous generation, Ernst Haeckel committed an act of fraud to prove that life had arisen spontaneously. He claimed to have discovered primitive cells that he named monerons. He had those on his tree of life there at the bottom. He described these monerons this way. He said, "They're not composed of any organs at all, but consist entirely of shapeless, simple, homogeneous matter. They're nothing more than a shapeless, mobile little lump of mucus or slime consisting of albuminous combinations of carbon." But where did Haeckel see these cells that he described as simple little lumps of mucus or slime? Nowhere. They were only seen in his imagination. They are a theoretical necessity for evolution, but not something that has ever been observed. But he made 30 drawings of these monerons, which along with 73 pages of what were nothing but speculations were published in a prestigious German scientific journal in 1868. He also gave them scientific names such as Protoamoeba primitiva, because a primitive protoamoeba must have existed before the complex amoeba we find today. So I guess he thought, why not just go ahead and make it up until it is discovered? Well, the extent of the detail that you see in these drawings of monerons is really a good measure of the fraud that he committed in claiming to have discovered these. Now, I'll point out that Ernst Haeckel was a professor at Jena University in Germany, the home of some of the finest optical equipment available of this day. He had no excuses for making such errors, but they weren't errors. These were just outright frauds. Perhaps a better example of the height of the deception is the fact that he also illustrated diatoms. Now, these are diatoms that you see here. Diatoms are single-celled algae that are enclosed within the cell walls made of silica, which have these beautiful geometric, symmetrical forms. Well, Ernst Haeckel published images of diatoms. Shown here, these are a couple of the plates that Ernst Haeckel made of his diatoms that he published in a book titled Art Forms in Nature. You'll see the German title on the bottom, but I can't pronunciate that, so I won't even try it. Haeckel again had access to some of the finest microscopes available of his day as a professor in Germany, and his work shows that he was well aware of the complexity of cellular forms. These single-cell organisms, these diatoms, are far from shapeless, simple, homogeneous lumps of mucus. And it isn't amazing that someone can see so keenly art forms present in nature, but not recognize what that implies. That behind art forms, there's an artist. Behind designs, there's a designer. See, God put evidence in the world that it was created, not just in the forms of in engineering or in designs, but also art is there to show that he is real. Remember, we were reminded in Romans 1:20 by Paul that God created the world to be such a way that it would witness to its own creation. And he said, "For since the creation of the world, his invisible attributes, his eternal power and divine nature have been clearly seen, being understood through what has been made, so that they are without excuse." So he put his own internal attributes within the creation to be a witness to us. But then he continues on. For even though they knew God, they did not honor him as God or give thanks, but they became futile in their speculations, and their foolish heart was darkened. Professing to be wise, they became fools. Well, that is the nature of science today, shackled as it is by naturalism. They simply refuse to acknowledge the truth of what they see. They can see complexity, they can see design, they can see art forms, but refuse to acknowledge it. Well, the truth is those Monerians that Haeckel imagined did not then and do not now exist. The actual organisms, the bacteria that were later assigned to this taxa name, Monerians, are in fact in possession of one of the most complex molecular machines we know of, the bacterial flagellum. They have an outboard motor, far from shapeless little lumps of mucus or slime. When I was in school, when I was in college, the bacterial kingdom was named the Kingdom Monera, an homage, I believe, to Ernst Haeckel for all the work that he has done. They were later renamed in eubacteria or archaebacteria, but originally named the Kingdom Monera. But far from shapeless little lumps of mucus or slime, bacteria are incredibly complex. So to reiterate this, after proposing this evolutionary tree, Ernst Haeckel subsequently fabricated both the top and the bottom. The fact is, those proposed organisms, that first abiogenetic cell, and that missing link between apes and humans never existed. They are complete fiction. And spontaneous generation theories were and are today something that has almost zero direct supporting evidence. The view has been upheld from the beginning for purely philosophical, religious reasons. Franklin Harold, shown here as a professor of microbiology at the University of Washington, and states this unequivocally in his book, The Way of the Cell. He says, life arose here on Earth from inanimate matter by some kind of evolutionary process about 4 billion years ago. This is not a statement of demonstrable fact, but an assumption almost universally shared by specialists as well as scientists in general. He says it is not supported by any direct evidence, nor is it likely to be, but it is consistent with the evidence we do have. But ladies and gentlemen, the only evidence is that life exists. And because evolutionists believe there is no God, it must have evolved spontaneously. Klaus Doss is the director of the Institute for Biochemistry at Johannes Gutenberg University in West Germany, and is considered today to be one of the foremost experts in the area of molecular evolution. He states this: more than 30 years of experimentation on the origin of life in the fields of chemical and molecular evolution have led to a better perception of the immensity of the problem of the origin of life on Earth rather than to its solution. At present, he says, all discussions on principal theories and experiments in the field either end in stalemate or in a confession of ignorance. George Wald, shown here, was awarded the 1967 Nobel Prize in Physiology and Medicine for his discoveries concerning the visual processes in the eye, and he openly admitted to a belief in the impossible that life arose spontaneously because he could not accept supernatural creation. In this article, Wald went on to write that the hero of the origin of life was time. Since life had billions of years to develop, what we would consider impossible is virtually certain, he says. And that, quote, time performs the miracle. He furthermore expressed the view that it was just a matter of time before RNA, DNA, and proteins would be created in a laboratory by simulating ancient Earth conditions. Wald's expectations, however, were never met by scientists despite an enormity of effort. The cell, as we know it today, is tremendously complex, much more complex than anyone in Darwin's day could have imagined. This is well illustrated by the microscopic organisms, those single-cell organisms that can move around because they have outboard motors like that possessed by the flagellum, shown here on this, also on this diagram. However, diagrams of a cell like this one are very simplistic because they must be due to the enormity of content and activity found within. The reality is a far cry from the simplistic diagrams that frequent textbooks. This image was produced by the Harvard Medical School, is one of the most detailed illustrations of a human cell to date. which gives us a better idea of just how complex the cell really is. To describe this complexity is difficult. Drawing an analogy for comparison from our experiences to illustrate how complex a cell is difficult because nothing in human experience is as complex as a cell. Our marvels of modern technology pale in comparison. The cell contains orders of magnitude more moving parts than a big jet airplane. Even a Boeing airplane factory does not even come close to the complexity we find in the microscopic world. The cell is truly more complex than an entire city, an entire metropolis like Gotham, or, well, this is actually Seattle shown there. A city is not even itself a fully adequate comparison, since it cannot replicate itself within 20 minutes or move from one place to another like single-cell organisms can. But there are a number of interesting parallels we can draw with the architecture and engineering found in a city. A cell has countless molecular machines. Some of these we call enzymes. It has structural support, like our bridges and girders in our buildings. It has factories full of machines. In fact, we call these organelles, like the mitochondria or the chloroplast. It has a central library of information and a system to distribute that information when needed. It has highways that assemble and disassemble, and armies of delivery vehicles that carry manufactured goods where needed, like the kinesin shown here. Truly, the cell is unparalleled in its complexity and design. Well, with such complexity realized, how do evolutionists propose that something that incredibly complex formed without a creator or intelligent design? Well, all they have is chance and the natural laws of molecular interaction, what they call necessity. So theories typically invoke a combination of these. Although the first cell is thought to be a very primitive ancestor of the cells alive today, experts agree that a living cell must be able to do these three things. These are the minimum requirements for any living system by experts. It must be able to store information, process energy, and reproduce itself or replicate. And that is quite a lot to be able to do. So let's walk through the steps that are proposed by evolutionists for the spontaneous formation of that first cell that is able to do these things. First, a cell must, for the first thing that must happen is what's called molecular evolution. Molecular evolution must take place. They argue that chance and the natural laws of molecular interaction caused chemical complexity to increase over time. And through this process, the molecular building blocks important for cells, like amino acids and nucleotides, formed in a theoretical primordial soup. Then these building blocks spontaneously join to form the macromolecules that are present in cells, most of which are big polymers like DNA or RNA or your lipids. Some assert a role of natural selection in the beginning stages of molecular evolution, although selection would not be possible until a replicating molecule or a cell existed to allow for natural selection. And of course, one requirement is lots of time. Then after you get those macromolecules would have aggregated together and formed the first cell. It is believed that a bacteria was probably the first cell. It's argued that a chemotroph was likely the first organism, an organism capable of using inorganic molecules as energy sources. Well, the origin of the complex organelles found in eukaryotes, like the mitochondria and chloroplast, is believed to be due to a process called endosymbiosis. That one cell consumed another cell without digesting it, allowing it to become an internal component. This is the theory called endosymbiosis. The mitochondria and the chloroplast are thought to have originated in this way. That one cell ate another cell, but instead of digesting it, it became a mitochondria over time. Well, shown here in this video, I'm going to run this for you again. You can see, I'm going to try to run this for you again. You can see these cells ingesting algae. Well, it ingests this algae, but it doesn't digest it. Allows the algae to actually live inside of it. The algae performs photosynthesis, which benefits this cell called a paramecium. Many organisms do this. It's what's called endosymbiosis. Sea anemones do it, coral do it. That's why they live in shallow waters. They ingest algae. The algae performs photosynthesis inside these organisms, and that benefits the sea anemone or the coral. This is a process that does happen. And they argue that through such an endosymbiotic process like this, these organelles first develop. Well, then, after you get a eukaryotic cell that has these organisms, you have to get a multicellular organism. Well, evolutionists argue that multicellular organisms could have evolved through what's called colonial aggregation. They point to colonial organisms like this one called a slime mold, wherein single-cell organisms, the ones you see here, aggregate together to form a single mass reminiscent of a multicellular organism. You can see it, what's called a slug, oozing along there. A bunch of those single-cell organisms aggregated together to form this slug. The slug will then at some point find a suitable location after it develops, find a suitable location, and form what's like a reproductive stalk. And it forms spores, and it sends those spores flying. But multicellular organisms are far from aggregates of single-cell organisms. They're made up of many different specialized cell types, like muscle cells, nerve cells, sensory cells, etc. Humans are composed of an estimated 100 trillion cells of many different complex cell types. Well, although endosymbiotic relationships do exist and colonial organisms do form from single-cell aggregates, these processes occur, I should note, in living populations. What evidentiary support do evolutionists have for the spontaneous formation of that first cell? What about the origin of the first macromolecules like proteins or DNA? The truth is nothing. They cannot even offer a credible method for the origin of the building blocks like amino acids that are used to form proteins, or nucleotides that are used to form the nucleic acids like DNA and RNA. They have nothing to use to explain the origin of these things. Well, let me try to compound the case against abiogenesis or spontaneous generation a little better by introducing you to a few of the big enigmas. Well, one of the most spectacular discoveries of our generation is that information governs the biological world. Biological information exists in the cells of all organisms as this complex molecule we call DNA, which stands for deoxyribonucleic acid. Well, this molecule, DNA, carries complex coded instructions for making an organism. Some DNA contains the instructions for the assembly and use of proteins in cells. However, only about 2% of genomic DNA contains instructions for making proteins. The rest serves various other functions, like regulating the countless biological processes that are involved with making an organism, development, adaptation, and even gene expression. Well, today, everyone acknowledges that DNA is information. This is not a contested assertion. Even Bill Gates said that DNA is by far the most sophisticated program around. And we're just now really beginning to understand what really exists there within genomic DNA. Well, the fact that DNA is coded information causes it to stand as the single most powerful argument for intelligent design that exists. Because in all past human experience, information has been seen invariably to originate from only one source, an intelligent mind. When it was discovered that information governed processes in the biological world, that discovery should have led to the conclusion that DNA was also the result of an intelligent mind, because that is how science is supposed to work. You're supposed to use your past knowledge to inform your observations and reach conclusions. In all past human experience, we've only seen information come from an intelligent mind. When we found information there governing biological processes, it should have led to the conclusion, or at least the hypothesis, that information also had an intelligent mind for its origin, but today, science is dedicated to explaining away all examples of intelligent design as originating through nothing but natural processes. Well, since intelligence is the only known cause of specified information, the presence of such information-bearing sequences of biological systems does point definitively to a designing intelligence behind life on Earth. Well, the amount of information packed into each cell, each human cell, is mind-boggling. And I want to try to make this point. Now, the DNA molecule is what you see there spinning on this graphic. The DNA molecule looks like a ladder that's been twisted into a shape that we call the double helix. Well, each step or rung, if you want to use the technical term on a ladder, for the step on a ladder, each step of that ladder is made up of one of 4 possible nucleotides that we abbreviate with the letters A, T, C, and G. Adenosine triphosphate, thymidine triphosphate, cytidine triphosphate, guanidine triphosphate. Thus, we abbreviate those with the letters A, T, C, and G. What you see on the right is DNA sequencing information. It's a big sequence of A, T, C's, and G's. Well, if each of those letters can be taken to be equivalent to the letters in our alphabet, then the amount of information in one human cell is approximately equivalent to that found in 1,000 books. Letter per letter. There's as many letters in one human cell, many of those nucleotides in one human cell as you would find in 1,000 books. Or to put that another way, a little pinhead-sized pile of DNA has as much information as you would find in 500 stacks of books reaching the moon, or a single stack of books reaching the sun, 93 million miles high. That is a staggering amount of information indeed. And where do evolutionists believe all of this information came from? Well, random chance processes is all they have, called mutations. They argue that random changes to the genetic code produced more code. Well, this assertion is one of the most ludicrous aspects of the theory of evolution. It's an affront to reason, it's an affront to logic, and it's an affront to common sense to propose that you could randomly change code and produce more code. Everyone knows that if you randomly change the letters in a book, you're not going to produce a second edition of a book, a better edition of a book, an upgrade to that book. You're going to destroy the book. If you randomly change computer code, you're not going to produce better computer code, you're going to destroy the code. Everyone knows this. It is ludicrous to propose otherwise. To propose such a thing is truly ludicrous. The theory is nothing short of science fiction. Mutations do not create improvements any more than they would make us into superheroes. Mutations destroy the genetic code and create disease and disorder. That's what they do. Well, only a scientist committed to a false worldview like naturalism would propose something they know is completely contradictory to our observations and experience. Francis Crick, for example, was the co-discoverer of the DNA helix. He won the Nobel Prize in 1962 for working out the shape of the DNA helix. He says this: biologists must constantly keep in mind that what they see was not designed, but rather evolved. See a person committed to their worldview, in this case, the worldview of naturalism, that natural processes alone were responsible for everything we see. Only a person committed to that worldview would make such a statement. That you have to not just remind yourself that what you see was not designed, but you have to constantly remind yourself that what you see was not designed, but rather evolved. This is what they have to do. There's overwhelming evidence for creation. God's invisible qualities, His eternal power and divine nature, clearly seen in what has been made. He made the world full of abundant evidence that it was made to be a witness to everyone, so they are without excuse. Professing to be wise, they've indeed become fools, constantly having to remind themselves that what they see was not designed but rather evolved. Well, this refusal by atheistic natural scientists to acknowledge the obvious implications of what information governing the biological world really means has led to assumptions that have impeded scientific progress in molecular genetics. For example, for decades, The portions of our DNA that do not code for protein production, which is actually about 98%, only about 2% of the genomic DNA, of the DNA in your cells, actually codes for the making proteins. The rest is mostly regulatory. But when this was discovered, it was assumed that 98% of our genome was just junk DNA, useless leftovers from our supposed evolutionary history. Even Francis Crick called it little more than junk. That 98% of the genome, called it a little more than junk. Well, this phrase, junk DNA, has haunted human genetics ever since it was first coined and has stagnated scientific research because many would not even bother to research the non-coding junk regions. Why research a bunch of junk? Why would you do that? And this has, and many have used our lack of knowledge about these portions of the genome to argue against intelligent design. For example, this is Kenneth Miller, a former professor at Brown University, and he was the author of Prentice Hall Biology, which is today the most widely used biology textbook used in U.S. public schools. He stated this in his article, Life's Grand Design. He said the human genome is littered with pseudogenes, gene fragments, orphan genes, and junk DNA, and so many repeated copies of pointless DNA sequence that it cannot be attributed to anything that resembles intelligent design. In fact, the genome resembles nothing so much as a hodgepodge of borrowed, copied, and mutated and discarded sequences and commands that has been cobbled together by millions of years of trial and error against the relentless test of survival. It works, and it works brilliantly, not because of intelligent design, but because of the great blind power of natural selection. Kenneth Miller. However, this whole concept of junk DNA is now fully dead. A massive research project called ENCODE, which stands for the Encyclopedia of DNA Elements, began studying the non-protein coding portions of the DNA. And in 2003, they began this massive project in 2003, a massive project that consisted of 400 scientists in 32 different laboratories around the world, and they released their findings over the course of several years, beginning in 2007. In 2012, ENCODE researchers estimated that 80% of the genome is in fact functional in the sense that it has specific biochemical activity. ENCODE scientist Tom Gigeris said this: almost every nucleotide is associated with a function of some sort or another, and we now know where they are, what binds to them, and their associations, what their associations are, and more. Ewan Birney, another ENCODE lead analyst coordinator, said it's likely that 80% will go to 100%. That pretty soon we will know what 100% of those nucleotides do, of that information does. And then he ultimately concluded this: we are the most complex things we know about. It's not surprising that the manual is huge. I think it's going to take this century to fill in all the details. Well, since ENCODE published its findings, other researchers around the world have continued to research the non-protein coding regions of the genome. Numerous publications can be found online. This was just one of them. In 2021, researchers published some new findings on regulatory functions of the sequences previously viewed as junk. In the Journal of Genome Biology and Evolution, they stated this: the days of junk DNA are over. When we studied genetics at university in the 1980s, the common doctrine was that the non-protein coding parts of the eukaryotic genome consisted of interspersed, useless sequences, often organized into repetitive elements like satellite DNA. Since then, however, the common understanding of such sequences has fundamentally changed, revealing a plethora of regulatory sequences, non-coding RNAs, and sequences that play a role in chromosomal and nuclear structure. The concept of junk DNA is now completely dead. Well, there's also a major enigma related to the protein machines that have been discovered in cells. And this is another component of the cell that evolutionists must account for in their theories related to the origin of life. In The Origin of Species, Darwin said this: "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. But I can find out no such case." He didn't say it would break down or it might suffer a little bit of harm. He said if we could find something that could not be formed by numerous successive slight modifications, my theory would absolutely break down. Well, we have found out many such things. And these, in particular, one of the best examples of these are these molecular machines, like the kinesin shown here again, or the flagellum mentioned previously. These molecular machines abound in the city of the cell. The kinesin shown there is a nanoscale micro robot carrying a vesicle of manufactured goods down one of the highways found in the cell. Machines like the kinesin are irreducibly complex. They simply cannot be built by numerous successive slight modifications. In his book, Darwin's Black Box, Michael Behe described irreducible complexity like this. He said, "Irreducible complexity is a single system which is composed of several interacting parts that contribute to the basic function, and where the removal of any one of those parts causes the system to effectively cease functioning." Well, protein machines like the kinesin defy natural explanation for their origin because, like a simple mousetrap, they have multiple parts that must be in place for the machine to work. Darwinian mechanisms require that such constructions be functional at every stage in their development through numerous successive slight modifications. Molecular machines will not function unless every single part are there, and they're there at the exact same time. They're assembled, all those parts are assembled together, and then energy is applied to them. Behe continues, "An irreducibly complex system cannot be produced gradually by slight successive modifications of a precursor system, since any precursor to an irreducibly complex system is by definition non-functional." Well, let me explain a little more what proteins really are. Proteins are chains of building blocks called amino acids. These chains, if you'll look at this diagram on the right, these chains must first be assembled from those amino acids. Then that chain of amino acids must be folded into various shapes, like the beta pleated sheet and the alpha helices. And then those must be assembled to form a protein subunit. That subunit must then be combined with other subunits before they are functional proteins. And machines like the ATP synthase shown here, one of the machines in the cell, this is one of the machines that actually converts glucose into ATP, an important chemical process called cellular respiration. This protein machine is made up of 500 separate protein subunits. Proteins are tremendously complex. These molecular machines are tremendously complex. And what have evolutionists today that can account for the origin of such things, the origin of these proteins? Well, the most famous experiment that attempted to solve the mystery of the origin of proteins was conducted in 1950 by Stanley Miller and Harold Urey. They sought to demonstrate how the building blocks of proteins, those amino acids, could form under natural conditions by creating a simulated theoretical atmosphere and a water cycle of the early Earth. Their apparatus was equipped with a port where they could input materials. They input methane as a carbon source. They input ammonia as a nitrogen source. They inputted some hydrogen and some water vapor. A chamber was added to supply some sparks of electricity. A condenser was included to promote chemical reactions, and a collector on the bottom where they could collect what had been made in the apparatus. And they succeeded in isolating about 2% amino acids from what was mostly toxic sludge, but they did isolate some amino acids. Well, there are several problems with the Miller-Urey experiment that are really noted by advocates of a spontaneous generation. They were supposed to be creating a model or reverse early atmosphere. But one main component of our gases was intentionally excluded. Can you identify what that was? They have methane, they have ammonia, they have hydrogen gas and some water vapor, but they excluded something very common in our atmosphere with intent. Oxygen. They excluded oxygen from this, and they didn't even include it as carbon dioxide. The reason for this is well known. Oxygen itself is a powerful oxidizer, which are chemicals that strip electrons off of other molecules. It's an oxidizing agent. And because of this, oxygen will break down amino acids. So any good biochemist who knows that amino acids will not form in the presence of oxygen. And it was assumed early on then that the Earth's early atmosphere must have been a reducing atmosphere, an atmosphere that did not have oxygen that would allow for amino acids to form. This was assumed by early origin of life theorists. However, it has been conclusively shown all the way back since the 1980s that the Earth has not had a reducing atmosphere, one that did not have oxygen, due to the presence of oxide minerals in even the deepest of Earth's rocks. From the Bulletin of American Meteorological Society in 1982, they say geologists know from their analysis of the oldest known rocks that the oxygen level of the early atmosphere had to be much higher than previously calculated. From the journal Geology in 1982, they say there is no scientific proof that the Earth ever had a non-oxygen atmosphere, such as evolutionists require. The Earth's oldest rocks contain evidence of being formed in an oxygen atmosphere. Well, despite this massive fundamental problem with the Miller-Urey experiment, the results of this experiment continue to appear in literally every biology textbook that discusses evolution. For example, Prentice Hall Biology that I mentioned previously, the author being Kenneth Miller, one of the most widely used biology textbooks used in U.S. schools today, says this. Over a few days, several amino acids, the building blocks of proteins, began to accumulate. Miller and Urey's experiment suggested how mixtures of organic compounds necessary for life could have arisen from simpler compounds present on the primitive Earth. But this is simply not true. The experiment did not produce anything resembling, quote, mixtures of organic compounds necessary for life. It is a well-known fact that the majority of the really important organic compounds, like proteins and DNA, have building blocks that exist in only one of their two possible forms. The experiment produced a racemic mixture of molecules, which is what we would expect in any kind of natural formation. However, again, the amino acids used to make proteins are only in the one form, the left-handed form or lever rotatory form. Also, the ribose, the sugar that's in DNA and RNA, only exists in the right-handed form. Well, numerous theories have been offered over the years in an attempt to explain how a pool of molecules could have formed naturally from that favor one form over the other. The most widely accepted theory is that they were formed on charged surfaces like clays or crystals. But experiments with these kind of substances have only been able to shift the percentages slightly in one direction. What they must account for is how only left-handed amino acids could form, or only right-handed riboses. Well, it is due to these challenges that the origin of life from space, termed astrobiology or exobiology, has become today NASA's main focus. That's why they're searching for water on planets and amino acids on meteorites and asteroids. In 2012, the 2012 discovery by NASA referenced on this slide at the bottom, you'll notice there, claimed to have found 4 times more of one type of amino acid on an asteroid than the other type. But again, proteins do not have just more left-handed amino acids, and the riboses don't have just more right-handed riboses. The DNA and RNA don't have more right-handed riboses. Proteins have only left-handed amino acids, and DNA and RNA have only right-handed riboses. It's not the kind of thing that would form in a natural environment, period. Well, another major challenge with this experiment in the 1950s is that they obtained no chains or polymers of amino acids. Because amino acids react more readily with other molecules than amino acids, a chain of amino acids forming under natural conditions is highly unlikely. And remember, proteins are chains of amino acids, we call these peptides, that must first be folded and combined with other peptides to form protein subunits, and then those must be combined with other subunits before we actually have a functional machine. The probability of getting a single chain of amino acids forming by chance alone has been calculated to be 1 out of 10 to the 164th power. That's a trillion times smaller than the odds of finding a single specific particle amongst all the particles in the universe. The law of probability states that the occurrence of any event with a chance beyond 1 in 10 to the 50th power, a much smaller figure than we're dealing with here, is an event that we can state with certainty will never happen, no matter how much time is allotted, no matter how many conceivable opportunities could exist for the event to take place. And then also note that many proteins, like the enzymes, are much, much larger. The ATP synthase, shown again here, has more than 2,000 amino acids. RNA polymerase, which transcribes DNA into RNA, has more than 3,000. Well, Francis Crick, again, one of the co-discoverers of the structure of DNA, was honest enough about the probability of spontaneous protein formation to admit that it was not possible. He states this. He said, if a particular amino acid sequence was selected by chance, how rare an event would this be? The great majority of sequences can never have been synthesized at all, at any time. But wait, there's more. It's not just the existence of information that origin of life theories must explain, and the formation of the complex molecules like protein machines needed for cell function. Evolutionists must also explain the spontaneous development of the information processing system called gene expression that cells must have to utilize the genetic code. So let me explain how this works. I'm going to explain how gene expression works, at least the portion of gene expression that's involved with the production of proteins, which, remember, it's only comprises about 2% of your genomic DNA. Well, genes, the protein coding genes, these are coded instructions for making proteins, which involves a two-step process called transcription and translation. In transcription, the cell makes a copy of the gene and sends that copy, called the messenger RNA, out of the nucleus to an organelle called a ribosome. The ribosome then assembles on the messenger RNA and translates it 3 nucleotides at a time. These mini coding units are called codons. And this instructs the ribosome on the amino acid to use when making a protein. These are some of the genetic codes. For example, UGC is a genetic code for cysteine, amino acid cysteine. CUG is a genetic code for the amino acid leucine. AGU is the genetic code for cyranine, and CGA is the genetic code for alanine. So the DNA is a code that instructs the cell how to do many things, and one of those things is again, how to make proteins. Well, this is how the code works. The ribosome reads the gene, and when it comes across these coding units, these codons, it uses those to assemble together amino acids to make this protein chain. Well, I want you to watch this in an animation. This is going to show part of this process we call gene expression. So what we're doing right now is we're watching through this animation. We're zooming down inside of the nucleus, entering what's called this nuclear pore complex, and you see there the DNA helix in this animation. Well, the first thing that must happen before a gene can be transcribed, can be copied, is first the double helix has to be unwound. And this is done by an enzyme called DNA helicase. It first binds to the double helix, unwinds it so that the transcription complex, a complex of proteins, can assemble on one of those strands. Several different transcription-related proteins must assemble first, and then the big enzyme called RNA polymerase binds to the DNA strand and makes a copy of one of the DNA strands. This copy is a message from the nucleus that tells the cell we need to make a protein. That message, called the messenger RNA, is escorted out of the nucleus through the nuclear pore complex, where that copy of the gene, a copy of that piece of DNA, is assembled, has a ribosome assembled upon it. It first attaches to one half of the ribosome, shown there. The other half of the ribosome will then assemble onto it. And the ribosome then starts reading the genetic code. And it reads this genetic code again, 3 nucleotides at a time. Now, part of the mystery behind the enigma of gene expression has to do with what's happening right here. The molecule that you see coming up to the messenger RNA is called the transfer RNA. It also contains a genetic code that's 3 nucleotides long, and it has an amino acid on the other end. So on one end of the transfer RNA, there's a nucleotide codon with 3 nucleotides, and on the other side of the transfer RNA was the amino acid. This molecule, this transfer RNA molecule, is bringing amino acids to the ribosome, and those amino acids would be used by the ribosome to make a protein chain if the codon matches. Well, this is one of the big mysteries. There are numerous difficulties for origin of life theories in explaining the association between the genetic code and the proteins they produce. One involves this molecule called the transfer RNA, again shown in a diagrammatic form here. The transfer RNA's job is to deliver the amino acid to the ribosome, where it becomes part of the assembled protein. However, the problem facing origin of life theorists is that the amino acid that is being carried is on the opposite side of the molecule from the genetic codon. The chemical properties of the genetic code do not favor forming a bond with any specific amino acid. So what the origin of life theorists cannot explain how the genetic code came to represent the amino acid. The amino acid is on one side of the transfer RNA, the genetic code is on the complete opposite side of the transfer RNA, and at no time are they ever connected together. So how did this genetic code come to represent the amino acid that it now codes for? There's nothing to explain this mystery. In fact, the whole process of gene expression is now recognized to be so overwhelmingly complex that only a person completely blinded by their worldview could not see it as evidence of intelligent design. How did, for example, the coding system come into being? DNA is required to make protein, which again forms the armies of molecular machines and makes all the basic substances required for life. And proteins is required to make DNA. DNA is required to make protein. Protein is required to make DNA. Plus, DNA is required to make more DNA. Sir Karl Popper, shown here, is best known for his contributions to the scientific method known as Popperian falsification, which is a means of distinguishing fallacious theories. Popper asserted that scientists can never prove a theory to be true, but rather can only disprove a theory or falsify it. Popperian falsification has been used to attack the theories like string theory and multiverse, which cannot be empirically tested. Anyway, Karl Popper said this about the origin of the genetic code. He said, "What makes the origin of life and the genetic code a disturbing riddle is this. The genetic code is without any biological function unless it is translated, that is, unless it leads to the synthesis of the proteins whose structure is laid down by the code. But the machinery by which the cell translates the code consists of at least 50 macromolecular components which are themselves coded in the DNA. Thus, the code cannot be translated except by certain products of its translation. He then continues, this constitutes a baffling circle, a really vicious circle, it seems, for any attempt to form a model or theory of the genetics of the genetic code. Thus, he says, we may be faced with the possibility that the origin of life, like the origin of physics, becomes an impenetrable barrier to science and a residue to all attempts to reduce biology to chemistry and physics. Karl Popper. Well, to make this case a little stronger, I want to show you what the simplest cell is that we know. This is the simplest cell we know of. It's a cell called Mycoplasma genitalium. It is a parasitic bacteria that inhabits the human urinary tract. It is an obligate parasite. It can't live by itself. It has to live within a host. But even this simplest cell has 482 genes. By deleting genes from the Mycoplasma genome, researchers were able to reduce the required number of genes to 256. But again, I want to note that it is an obligate parasite. It has to live within a host. It can't live by itself. And again, this is the simplest cell we know of. It can live with 250 genes or so, which if we equate that to 250 proteins that it produced, means that this cell, remember the chance of, if we assume these proteins are simple proteins with only 150 amino acids, that means it's 250 required proteins. If we multiply that by the probability of one protein forming that's only 150 amino acids long, that would give us a probability of this cell forming, and not of everything it needs, just for its 250 proteins to form. If there are only 150 amino acids in length, that gives a probability of 1 out of 10 to the 41,000th power to just get its 250 simple proteins to form. Not its DNA, not the other macromolecules that are required for life, like carbohydrates or lipids. That's just to get its proteins to form. The fact is that life is far more than just a bunch of chemicals or even protein enzymes. If we put every single molecule together that is required for life, we still cannot reconstitute a cell. And again, Francis Crick, the co-discoverer of the DNA helix, reasoned that life could not have evolved from non-living chemicals under any conceivable conditions on Earth. He said this, an honest man, armed with all the knowledge available to us now, could only state that in some sense, the origin of life appears at the moment to be almost a miracle. So many are the conditions which would have had to have been satisfied to get it going. But, of course, to Francis Crick, the idea of a creator was unacceptable since it would go against his atheistic faith. Therefore, he proposed in his book, Life Itself, that primordial life came to Earth from space. Some believe that life might have arrived being carried by an asteroid, a theory known as panspermia. But Crick believes that life was shipped to Earth billions of years ago in spaceships, supposedly by more evolved, therefore more advanced alien beings. Francis Crick, the co-discoverer of the DNA helix. Well, Paul warned us of such things in his book to Timothy. He said, the time will come when men will not put up with sound doctrine. Instead, to suit their own desires, they will gather around them a great number of teachers to say what their itching ears want to hear. They will turn their ears away from the truth and turn aside to myths. Well, ladies and gentlemen, do not be led away by the false teachings of evolutionists. They indeed have turned their ears away from the truth and turned aside to myths. Myths like spontaneous generation, that myths that humans have evolved from apes. Scientists have no support for these theories that they teach today as though they're absolute facts. Truth is, we did not evolve by natural processes, but rather we are the very purpose of the creation. made by our loving Father in heaven to have fellowship with Him in both this life and the everlasting life to come. Let me close.